welcome to Turn the Page, the official podcast of the Syosset Public Library.
Hi, this is Jessica at Turn the Page podcast at Syosset Library, and I am here with
Linda Maria Frank.
And Linda has joined us as a lecturer. What is the name of your lecture series?
My lecture series is called Topics in Forensic Science, and really what I have done is I have taken my experience teaching forensic science at Seaford High School and also at Hofstra University, and I have picked the topics that I have found most interesting, and actually my audiences do too. So I have a cluster on DNA technology and some interesting stories about DNA, and then I have a cluster about various kinds of crimes, and now I'm branching out into trying to connect literature to actually to forensic science. I have one called Plants, Poisons, and Plots. So
That's extremely cool. So you were a teacher prior
yes,
to this.
I taught everything from 5th grade to graduate school. I just kept graduating with the kids, and actually that's how I got into the lecture circuit, because when you teach middle school and you teach high school, it's a very different kind of thing than lecturing. You're doing a lot of interactive, you're setting them loose, allowing them to really take control of their learning. But in a lecture, you're standing up there and you're a performer. So what really enabled me to think that I could do this was college teaching, because they plunked me into a lecture hall with 125 students, and that's what I had to do. And I really got to enjoy it after a while. So that really was what allowed me to do it. And then, of course, when I retired from teaching, and it's sort of not really just when I retired, I wrote a mystery series. I started the mystery series when I was still teaching high school, and then I put it aside. And then when I finally retired entirely from teaching, I picked it up and wrote 2 more books, so I have 4 in the series. And they're called Annie Tillery Mysteries, and my tagline is Nancy Drew meets CSI, because I loved Nancy Drew when I was a kid. She made me a lifetime reader, and I taught forensic science. So my books, and I'm very proud of them, they are 4 very wonderful stories, because I was able to, in my courses that I took to learn how to write, I was able to get that talent of a last page of a chapter pushes you right into the next chapter. And I also, because I'm a teacher, and I had a reviewer say this to me, how did you sneak so many facts into your books? And I said, I'm a teacher. So every book has history, which is not fiction, and science, which is not fiction. But of course, the story is fiction. So I'm very proud of them. And again, that was another entree into the world of lecturing, because at first I started with book talks, and then I found out that book talks were not really so popular. And so I said, well, let me take my forensic topics that I find fascinating and let me make lectures based on that. And that's how I got here.
And you had said before this that you were a reluctant reader. You were
Yes.
Nancy Drew was your...
Absolutely. I did not like to read. I still, as an adult, it's not that I don't like to read. It's that I can be very easily tempted into doing something else.
Yeah, that's, well, welcome
I'm
to the
very
world
kinetic.
of everything. Yes, absolutely. Because I was a reluctant reader as well, and here I am, a librarian. And for me, it was The Lion, the Witch, and the Wardrobe, which was my first step into the world of fantasy. And now I read a lot of different types of books, but I think we both agree it's about putting the right book in the
Yes.
hands of the child that's going to just
Absolutely.
spark that.
This is what I tell parents when I talk to parents, and I get a chance to do this because I do book events for Long Island children's writers and illustrators, and we do book fairs at night so that the parents can come, and the parents will say, my child just doesn't like to read, and I said, well, try this book because it's exciting.
Yeah.
So that's how you get them to read.
Absolutely. Without a doubt.
We can only hope.
So from Annie Tillery, and how many books are in the Annie Tillery series now?
4.
For.
Do you want the titles?
Yes, please.
The Madonna Ghost, which takes place on Fire Island. I can't tell you about the plot because it's a secret. The
That's
ghost
okay.
is hiding a secret.
That's okay, but the fact that you said that it's on Fire Island, we Long Islanders, as you know, anything that has to do with Long Island, we just eat it up because when people think of New York, they think of like this very small area, which is New York City.
That's right.
But there's like tons of space upstate. It's huge, and then there's Long Island. I think that's why we hold so fast to every lyric of a Billy Joel song that mentions anything on Long Island. It's like Oyster Bay. What? That's a place that I know.
Exactly. And Long Island has an incredible history.
It really does.
Because you had to bump into Long Island to get to the United States or to get to America when you were a colonist. So
yeah,
it has a rich history.
There's a lot of Revolutionary War history here.
Yes, absolutely.
So the Madonna Ghost, which takes place on Fire Island.
Yes, and then there's Girl with Pencil Drawing, which takes place in Brooklyn and Manhattan, and it's about art forgery, which is one of my favorite topics. That's one of my lectures, actually.
Oh, cool.
And then the third one is Secrets in the Fairy Chimneys. I must admit, it has nothing to do with fairies. Fairy chimneys are geological formations in Turkey, and it takes place at an archaeological dig, which is a real place in Turkey.
Awesome.
And it is a page turner. And then the fourth one, again, we go back to Long Island. The Mystery of the Lost Avenger. The Avenger is an airplane that was manufactured by Grumman during World War II.
My husband's grandfather flew one.
Well, you know, George Bush the Elder, that's the plane he crashed in.
Really?
Yes.
My husband's grandfather crashed. My grandfather and my husband's grandfather both crashed in planes, but I'm not sure about my grandfather's plane. But his was definitely an Avenger,
Yep.
and I did not know that. That's very wow. Okay, so a
Yep.
little presidential history as well.
So what I did in that book was we're going back to 1943 and the present. And what happens in 1943 is my main character, Annie Tillery, her great-grandmother is a test pilot and a WASP, which is Women's Air Service Pilot, for Grumman. And what happens is she isn't in the crash, but a plane is being flown by a WASP to the West Coast where the Avengers were put on aircraft carriers, and the plane crashes in 1943, but is not found until 2014 because it crashes in Appalachia. So when they find the plane, there's a note in the plane that traces back to Annie's family. And, of course, Annie has to solve this mystery. So we go back and forth between 1943 and the present. We go to NCIS headquarters in Washington, D.C. to solve this problem. We fly out of Republic Airport in a small plane that Annie's boyfriend has just gotten a license to fly. And it's all about Rosie the Riveter, and it's all about the sabotage that took place on Long Island during the war. It's really, it's a ghost story, it's a mystery, it's a romance.
That sounds awesome.
It's a wonderful book. I love that book. And the inspiration for that came one day. I was in the Cradle of Aviation Museum because I have to go there every time my son comes to visit. We have to go there, and I'm walking around and I see the Avenger, and there's a plaque in front of it about how the women did everything. And you could climb into the plane. So I climbed into the plane. I sat in the cockpit. I couldn't see over the cockpit, and I thought to myself, how do they fly these planes? And I said, I have to write the story about these women. And that was my inspiration.
That's so awesome. And yes, we go there frequently, and we're always climbing into that plane.
Yeah. I love that place. And,
Yeah.
and we are the cradle of aviation here.
We are, absolutely. Yeah, that's another thing that Long Island certainly has in their history is the whole aviation, history. And Grumman is right around the corner from here, less than 15 minutes up the road.
That's
and
right.
Roosevelt Field at one point was not just a huge mall.
Yeah, my dad actually watched Lindbergh take off from Roosevelt Field on his famous journey across. And I actually one of my lectures is the Lindbergh kidnapping.
Yes, I noticed that. So let's get into your lecture series. Here we are.
Okay, so we're really going to talk about my DNA cluster. Okay. And the reason why I got into this is because I actually, besides teaching forensic science, I also taught a course. It was called Biotechnology and the Bioethical Debate. And the reason why, and this is a course I designed myself, the reason why I did that was I found that so many people use the term DNA and have absolutely no idea what it means, how it works, and why it's useful. And also, I was getting into arguments with my friends about genetically modified food, and they were thinking that, the entire population is being poisoned. And actually, that's not the way it works. And whether whether you agree with it or not, you really have to understand how it works. So I decided, okay, I'm going to really get into this. You have to understand that the structure of DNA was figured out, and the two scientists who did it, who were called Watson and Crick. By the way, Watson was the director of the Cold Spring Harbor labs for a very long time. He's too old now. These two gentlemen Watson and Crick won the Nobel Prize in 1958. Okay, that's the year I graduated from high school. Everything I learned in biology, okay, it was all descriptive, but now we knew how things worked. And this was so fast. Everything I learned about DNA and had to teach in biology, I had to learn myself by going to workshops and whatnot. So this became like my crusade. I'm going to teach people about DNA. So I thought, how can I do this in a very interesting way? And then, of course, I bumped into teaching forensic science, and forensic science now uses DNA all the time. In fact, the first case on Long Island that used DNA as evidence happened in 1989, I believe. It was the murder of Kellyanne Tinez.
Oh.
She was a young 13-year-old girl who was murdered by a neighbor. He managed to entice her to come to his home because his younger brother was in her class. And she went, and she was never heard from alive again. And they found her body in the house, and they used DNA. Now, I watched the entire trial, and it was so confusing to the jurors because they brought in expert witnesses from Cold Spring Harbor labs and other places, and no one understood what they were talking about. This is all brand new stuff. So the jury convicted, but they convicted on bite marks, not DNA. So,
yeah.
Okay.
Even though it was, even though it was available to them, I suppose it just was, it wasn't, as you said, you learned a lot. Yeah.
It was very arcane at that time. And then little by little, DNA has been really, how shall I say it, it's been brought to the forefront. And people have become interested, and they have started to read about it. The other case where we know DNA really figured a lot was the O.J. Simpson case.
Yes.
A mountain of DNA evidence. And what happened in that trial was O.J.'s lawyers managed to discredit the expert witnesses, the technologist who collected the evidence, and actually tainted the head detective. They proved him to be a liar in one case, and that did it. But there was a mountain of DNA evidence there. So, but that's water under the bridge. So that's how I got into climbing the spiral staircase of DNA. And so would you like me to talk a little bit about what's in the lectures?
Sure, please do.
Okay, so the first lecture is really how DNA technology is used to solve crimes. And I like to start my lectures with an interesting story. So I start that one usually with the story of Sir Alec Jeffreys. Sir Alec Jeffreys was an Englishman from Leicester University in London, and he was doing medical research. Most of what we know about DNA and that we use in forensic science comes from medical research, because this is where we're trying to find out what is it in DNA that causes disease. So he was studying neurological problems, and he was looking for a DNA marker, because this is how DNA works. DNA is found in the nucleus of a cell, and what happens in the nucleus is the DNA replicates itself so that the cell can divide, but also at the same time, there is another molecule that's manufactured from DNA called RNA. And RNA is a messenger. It leaves the nucleus. DNA really can't leave the nucleus because the little holes in the nucleus aren't big enough, to make it simple. And the RNA goes to another part of the cell called a ribosome, and that's a protein factory. So the code in the DNA is really found in a gene, and one gene codes for one protein. And that's as simple as you can be. But your entire life is proteins. You're either structural proteins, hormones, or enzymes which control chemical reactions. So what scientists have found is they've located genes. That's fine. But what they have noticed is that the genes actually have markers on the DNA that precedes and follows the genes, and those markers can indicate whether or not you're going to have a disease or not.
I've been hearing a lot about this lately. This has been very, very popular in the news lately.
Mm-hmm. Yes, and people are really, little by little, the general population is becoming quite conversant in how DNA works, and of course, everyone wants a cure for cancer or any other disease that comes up. The genetic diseases in particular, we want a cure for them. So the research is heavily into that. And we're finding out that actually we can treat cancer with manipulative technologies with DNA. And we've also come up with a tool called CRISPR. And CRISPR is CRISPR. And what it is it's a little chunk of RNA combined with an enzyme, and the enzyme acts like a scissor. And the RNA, you have to choose your RNA so that, because RNA will latch on to DNA. It's like Velcro. Okay?
Huh.
And so if you choose your RNA to go to a specific location on a DNA molecule, the enzyme then can act as a scissor and either cut out a piece of DNA, which is causing a problem, or insert.
So this is really interesting because this is why people are starting to call it gene editing, I think, has been the term that I've been hearing a lot.
And designer children.
That is another thing. Are you familiar with there was a movie way back in the 90s called Gattaca. Gattaca. Yes.
I use Gattaca all the time because people say, well, some people will say every child who was born, we should take a DNA sample and keep it on file. And then there are people who say, oh, sure, that's a really good idea because insurance companies will get a hold of that and they'll say, oh, you have a proclivity to develop heart problems or some other disease, diabetes, and so we're not going to insure you. And the Gattaca, I believe, really was based on how smart you are.
It was a good movie.
Oh,
I
yeah.
have to say, I really liked it, and I think about it a lot when I hear about these things like designer children and
every technology has its dangers. And people ask me when I do the lectures, people say, well, how do we control this? Or how do we prevent the negative aspects from happening? And I say, it's like everything else. We need to have oversight.
Right, of course.
And regulation.
Of course, because of course people are concerned about falling off the cliff into eugenics, which is which is something also that kind of always buzzes in the back of everybody's mind when they
everybody
think about
who thinks.
Everybody who thinks. Yes, thank you. I mean, it's certainly because of course you want your children to be happy, healthy. You don't want there to be like a surprise tragedy.
Exactly, sure.
But there is that other side to it, and I think you bring up a very good point about oversight.
Yes, well, and then oversight has to be oversighted too. So it's just, and people don't really think about science. They think about science as things are invented, okay? And really, science is a process that has no how shall I say it, no prejudice as to what the results of their investigations are. Science is a process of investigation to try to explain the natural law, and it should have no prejudice, left or right or middle or whatever. Whatever comes up is what comes up. And,
right.
And people think that science has its own mission, so to speak, and its only mission is to find the truth. So
right.
But scientists are human.
Yes, they are.
Okay.
Yes.
So we have to be careful about that.
Well, anyway, that's been very interesting. So anyway,
these are the questions. I mean, I'll talk about the technology, but it's the people in the lectures who come up with these questions.
Right, of course.
And I have to be prepared to answer that. I mean, some of it I will say, this is my own opinion about this.
Right.
And I mean, my ongoing fight with a very good friend of mine about genetically modified food has become actually, to my mind, rather amusing. But Not to her. Okay, so that's I explained DNA technology, and I explain the uses for criminology. So there's actually 2 tests. One of them is called restriction length fragment polymorphism, and this is everything in DNA technology is letters. It's alphabet soup. So it's RFLP, and that is an extensive search of your DNA, but everyone thinks that if your DNA is analyzed, they analyze your entire DNA. Your entire DNA goes around the world a few times if you stretch it out.
Yes.
It isn't. It actually is just looking for little chunks of DNA that are highly researched so that we know what we're looking at, because what happens is we're not looking at genes. We're looking at those traffic signals and markers I talked about. And what scientists have found is that in the human population, those markers will occur just like blood type in specific percentages in the population. So let's say you have type A blood. That means you represent 50% of the population. That's not really helpful in criminology. So then we have to look at something else. Well, let's look at your Rh factor. And then now let's look at your MNN factor. Let's look at your blood enzymes, blah, 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 all the way down the line. Well, the same thing is true with RFLP. We keep looking at different markers, and every marker represents a percentage of the population. And you know what happens when you have 1 out of 10, and then 1 out of 4, and then 1 out of 200. The denominator keeps getting bigger.
It's like, yeah.
And so the possibility of you being that person who committed that crime.
It's really interesting because I'm picturing, as you're talking about it, the old lineups, where somebody's behind the one-way mirror, I should say. But it's almost like a lineup with your genetics.
Yes, it
your
is.
DNA. That's really interesting.
That's a good way. That's an excellent way to put. I'm going to use that.
Oh, please do. Please do. Thank you. Because that's what's in my mind.
Yeah.
Instead of having the witness come in, it's a whole other one-way mirror.
Right. And the other technique is called PCR. And PCR stands for polymerase chain reaction. Means nothing really. But what it is it's a process whereby you can take a tiny amount of DNA and multiply it. It's like Xeroxing the DNA, and then you can analyze the DNA. And the analysis of the DNA is simpler than RFLP, but it's almost like the home pregnancy test. You're really sticking like a strip of paper that has little pieces of RNA on it or DNA, and the sample that you have analyzed from the criminal or from the evidence will stick to certain ones of those and give you a DNA profile. And it's used because it only takes a half an hour to do. I've taken my students to the lab at Cold Spring Harbor and also the one at Stony Brook, and in a half an hour you see the results. So it's used to exonerate, not to convict.
Oh, that's interesting.
Mm-hmm. The courts are very cautious about how they use evidence and what they will accept, and that's really good. There's a case called the Daubert case, which is the federal rules of evidence, which really says that you have to adhere to the same stringent codes that scientific research adheres to see whether or not your evidence matches up with your suspect, which is a good thing. It's a good thing.
I would say so. So you mentioned a little bit about those CSI shows. So
yeah.
They're definitely very entertaining. What would you like to say about them?
What I do with that one is I go through all of the divisions of the crime lab, and I talk about what they can actually find out, and then I'll match up a case. But as far as DNA is concerned, the one lecture that I have on DNA, which people love, is the 4 famous murder cases. And, of course, it's 4 women. So this, of course, women
in true crime. Oh, my God.
For some reason, people love it when women are murdered. It's more...
There's podcasts alone about true crime, and it's really very it's a phenomenon. That's all I'm going to say, because and I know several people on staff are huge fans of true crime, and since I've moved from children's to adults, I've been finding myself reading a lot more domestic thrillers as well.
Mm-hmm.
Yeah.
Well, I took 4 cases, and Two of them were, there was no DNA technology at the time, and that was Marilyn Shepard. Sam Shepard was the basis of the show The Fugitive, which ran for years. The bushy-haired intruder, okay?
The one-armed man, right?
Yes. And there was, I don't know how this man was ever convicted on the evidence they had, because there was a third blood type at the crime scene. That indicates that there was another person, and I don't know how he got, but his life was ruined. His life was absolutely ruined. So eventually what happened was, when DNA technology became useful and accepted by the courts, his son got a scientist and a lawyer and whatnot, and his father was already dead. But he was exonerated based on the DNA evidence. And the evidence is saved for a very long time. And DNA is pretty resilient depending upon how it's collected and preserved. And it was degraded. So the scientists who did the technology said that you could not eliminate that there was a third person there. So that was enough. The other old case was Mary Sullivan. Mary Sullivan, it was suspected that she was a victim of the Boston Strangler, okay? And this was in 1964, no DNA evidence. They did have semen that was found on her body and the blanket she was found wrapped up in. And Anthony DeSalvo, who was the Boston Strangler, was already in jail for some other crimes. There were 11 murders attributed to him, and he was never convicted of any of them. It was another rape, and he was murdered in jail by his fellow inmates. That's how nice he was. So what happened with him was they came up with another test called YSTR. And YSTR is taking little tiny chunks on the Y chromosome, which only belong to men. They only have that Y chromosome. So what they did was, he had a nephew, and they took, they asked him for a sample, and he said okay, and they looked at his Y chromosome because it would be inherited on the same line, and they compared it to the evidentiary evidence, and there was enough of a concordance that gave them the ability to exhume Anthony DeSalvo's body, and they took DNA from his teeth, and it was a match. So he did kill Mary Sullivan.
Oh, wow. That's...
Yeah.
Wow.
Yeah. That's pretty impressive. And then the other two cases were OJ, okay, and I don't know if you remember the Lacey Peterson case.
I do. That was...
Tragedy.
That was... Lacey Peterson was that the early 2000s?
Yeah. Mm-hmm.
Yeah, I definitely remember that.
2003,
Yeah.
I believe it was.
Yeah.
And I actually used that case as my final examination when I taught at Hofstra. And it was, she was 9 months pregnant, and she was murdered. She disappeared, supposedly. And they suspected they always suspect the husband. They always suspect someone who's close to you because you have more reason to kill them.
And that's also, I mean, why in kidnapping cases, I think they always say that a large percentage of it isn't, I mean, it's always unfortunate. There are cases where it's random, but a large percentage is somebody that the family always knows, if not someone who's related.
Yes. Oh, definitely. So they were watching him. His behavior was strange. They had his girlfriend wear a wire. He never really quite admitted it. And the interesting thing was, they used DNA because what happened was, they asked him where he was when Lacey disappeared, and he said he was fishing in San Francisco Bay, and he gave them an exact location. And so the prosecuting attorney, what a bright guy he was, he went to the tide and current charts, which every body of water has. And what he did was, he tried to figure out where a body dumped at the spot he admitted to be fishing at would wash up eventually, and how long it would take. Lo and behold, in I think it was 4 months, which is what he thought it would be, these remains washed up. They were hardly even bodies, but they were remains, and one was a fetus and one was a torso. They couldn't even tell if it was male or female, because if a body is dumped in the water, the fish take care of it pretty
Yeah.
much. Fish and crabs and everything else. So, but what they did was they went to Lacey's parents and they took DNA samples and they ascertained that the DNA from the adult body was a child of the parents, and then they took the DNA from Scott and from the Lacey's body, and they ascertained that the fetus was the child of. So they had found Lacey and the child. Okay, so that's how they used the DNA to identify the bodies. But they didn't use it to convict him. What he was convicted on was a lot of circumstantial evidence. I never thought they'd get a conviction on this because they had no hard physical evidence. They had one hair of hers on the boat, I think. She was married to him.
Yeah, she would have been on
Yeah.
that boat a lot, one would assume.
Yeah. Or even if she never went on the boat, he could have had her hair on his clothing and it would end up.
My hair ends up on my husband's sweaters all the time.
Yes. This is Locard's principle.
Yeah, there's without a doubt. I mean, my husband could have a brand new car that I haven't been in yet, and my hair is on his shoe,
Or his
or
coat
yeah,
or sweater,
exactly.
and then it's on the seat of the car. Yep.
With a lot of cat hair as well. And
Me too. Would you like to? No. I know. So that was and this lecture is very popular. Then I went on to my favorite, which is solving ancient mysteries using DNA. Okay, and I start with the first ancient mystery, which is ourselves. So I go into how Ancestry.com works. And people, my audience is many times primarily senior citizens because I do continuing education at Stony Brook, Farmingdale, and Malloy. And so they've all done it, because they have the time to do it.
right.
And so I explain how it works and, what happens when you spit into the tube. Okay, so the spit is analyzed in 3 ways. They look at 3 kinds of DNA. One of them is mitochondrial DNA. Mitochondria are little structures in the cell that are not in the nucleus but contain DNA, and they're responsible for converting sugar into energy. And mitochondrial DNA is simpler. Everyone has it because it is inherited through the maternal line. Okay. And then they take Y DNA, YSTRs, again, only for men. And then they take what's called AT-DNA, which stands for autosomal DNA. And this is 700,000 little mutations on all of your DNA. So that's a lot of DNA. And really what they're looking at only is something called SNPs. And SNP stands for single nucleotide polymorphism.
That's adorable, though.
SNPs. And they're actually little mutations that occur in all of our DNA. And again, the mutations that occur represent a specific percentage in the human population. So what's happening with Ancestry and 23andMe and Heritage and all the other companies is they are amassing this enormous database. Okay, because you signed the paper. Oh, sure, you could put my stuff in the database. And now people are saying, I don't know, because you know what's happening? They are using these databases to check evidence from crimes to see if they can find not the perpetrator, but a relative of the perpetrator.
I've heard that.
Yeah. So, it's interesting and it's a great tool. But again, what are we looking at here? We really have to think about these kinds of things. So after they get all of these 700,000 SNPs and mitochondrial and Y, then they do a mathematical calculation and you get the pie chart. And why can they say this belongs to a specific geographic region? Because geographic regions, people who live there tend to be isolated in that region and intermarry, and so there is a collection of mutations that represents that population. They can actually find you. I don't know why this is so. I can make a guess, but they can locate you to a town in Ireland. I think it's a lot of Irish people must do this. Okay, so and also it's beginning also in Italy as well. So. There are 153 what are called haplotypes. The term haplotype is that pie chart for you. So, I think that's fascinating. I think it's
It is.
wonderful.
It is really interesting.
Did you do it?
Not yet. I want to. I'm not sure which what is it like? Which company to go with?
Well, if you
23andMe was very popular in this library. I will say a few of us have done that, but I'm not sure.
Well, 23andMe also gives you your profile, your disease profile, and Ancestry does not. But Ancestry also allows you to go into all of the, build a family tree. I did Ancestry, and the amusing thing about me is that, well, I'm Polish, okay, on both sides. All right, so I thought, well, yeah, but the French tramped through Poland. The Russians tramped through Poland. Poland wasn't really Poland for a while. The Germans. I
Yeah.
thought I'm going to have this rich, and I found out that I'm Polish.
I am a quarter Polish as well, so we might have
We may be relatives.
we might have relatives, common relatives.
It's funny. So that was. But then my son, because his father, we thought was just Spanish and Italian. My son was 2/3, I believe, Eastern European, and then all the rest was all over the world. So evidently his father's ancestors traveled a lot. But it's fascinating. So that was the first ancient mystery. And then in that lecture, I go into a lot of stuff. In fact, it's now 2 lectures. I also, after talking about what a haplotype is, go into the story of the Kohanim. Now, I always get this wrong. The first Jewish priest was anointed by Aaron, Moses' brother, and that was a Cohen.
And that's why they say that if you have the last name Cohen, you are related to Aaron, right? Correct? Yes.
Yes, and so there was a Jewish, again, a scientist doing research, and he decided his name was. Let me see if I can remember this. Koreki was his name, and what he was looking at was all these Cohens who don't look alike, and he's saying to himself, how can they all be related? So what he did was he went and he took samples from Cohens from Ashkenazi, from Sephardic Jews, from Jews all over the world, and he made a haplotype, and guess what? They are related.
Wow, that was not the ending I was expecting to that
And
story.
Even better is there is a tribe in South Africa called the Lemba,
Yes.
and they wear yarmulkes and they wear the stole and they carry out Jewish rituals. And he did their DNA profile, and they are related. They may be the lost tribe.
Yeah, that's very very interesting as well.
Yeah.
I read about them a long time ago, and I'm forever educating people about their existence.
Mm-hmm. Oh, it's fascinating. Well, I mean, it makes sense. It's Africa, right?
Absolutely. And people don't necessarily think of that connection, but it does make sense.
Sure, because Israel is really
Africa.
in Africa.
Yes.
Okay, and so that's another topic that I go into is human migration, and National Geographic did a project a while ago about how people migrated from the Aldovai Gorge, which is where the human population started. And so we talk about that a little bit. I go into finding the mitochondrial Eve, and that's basically based on the fact that mutations are like clocks. So the more mutations you have in your DNA, the older you are. So they, again, took samples from women all around the world, and they found that African women had the most mutations. So they're the oldest, so they must be the origin. And I go into Jurassic Park because isn't it a great idea to reproduce dinosaurs?
It's too bad that my co-host and colleague Stacy is not down for this, because if you ever want to come back and just talk about that, she is here for it. That's like one of her. She loves Jurassic Park.
Well, actually, the science behind it makes sense and it's logical. And there was a Russian scientist in Siberia who actually found a frozen woolly mammoth.
I read about that.
In such good shape that it had buttercups in its mouth still. And he said, let me take the DNA and do the same thing they did in Jurassic Park, put the DNA in an egg cell and let it gestate in a modern cow and produce a woolly mammoth. That's another thing we need is woolly mammoths.
I mean, they're kind of adorable
But-
and scary.
but you know, the good thing that they could do with this is they could preserve um, species that are going extinct. And if they could actually find some DNA from extinct species, then perhaps they could bring them back or at least preserve them in some way.
I think that's very interesting and very good use of the
Sure.
science.
Sure.
For sure.
Sure. Yeah, well, this is the whole idea of, science and technology are used for good and bad. So,
Yes.
but I love to make a joke. I love to do the Jurassic Park one, and I'll go through the whole technology, and I'll say, isn't it a good idea to have dinosaurs? Don't we have enough problems?
It worked. It worked really well for them, but not really. No.
No. I mean, the only way we got rid of them was a big meteor hitting the Earth, so.
That was the first time. The second time, Jurassic Park spawned
Yeah.
so many sequel books and movies.
Yeah, yeah. That was a great movie, though. So then I go into the ice mummy. Do you know about the ice
I
mummy?
do.
All right, Otzi is his name. And he's called Otzi because they actually, doing scientific testing, located the original area of the Tyrolean Alps where he came from. And it's a town called Otzital. So Otzi. Now, the whole thing with Otzi, his body is frozen in Italy, because they finally determined that he was found in Italy. But they used that copy machine that makes a plastic replica, and they made a replica of Otzi. They painted him up, and they sent him to Cold Spring Harbor. So he was in the Cold Spring Harbor for a while.
He was right here on Long Island, folks.
So what happened with Otzi, though, is they used everything. They used a branch of science called taphonomy. And taphonomy is a branch of science that tries to determine the circumstances of a person's death from the remains that they find. Now, Otzi was really well preserved. And I have a lot of fun with this one, too, because I say that two hikers came across parts of a body that were sticking out of the ice. They were hiking in the Tyrolean Alps. And so they called the police because they thought it was probably a hiker who died in a snowstorm, blah, blah, blah. The police came and they started chipping him out, and they noticed that his wardrobe is not exactly LL Bean. It's a little different.
And he wasn't a lost hippie or anything like that.
So yeah, exactly. He was not a lost hippie. So they called in the archaeologists, and then the archaeologists more carefully removed his body, and they started using archaeology and anthropology and scientific testing. They dated him using carbon dating, and they found out he was 5,000 years old. And they figured out so much. And I'm not going to tell you everything because you have to come to the lecture. So I also talk a little bit about mummies. Mummies can be, the blood that they find, the dry blood in a mummy can actually be typed just by regular blood typing.
Oh.
People are amazed at that. And so you can also do, DNA on the mummies, and probably figure out from which mummies are in the London Museum still, and which are in New York and whatnot, who's related to who.
I think they have a few. Are they called bog babies in the London Museum as well?
Well, the bog people are, these are people who are preserved in the bogs in Northern Europe.
Right.
England and Northern Europe, because the bogs are made of what's called peat.
Yes.
We know peat as peat moss.
Yes.
But a real peat bog, this is like swampy.
Yeah.
And peat is the first stage in coal production. It's extremely acidic, and these people, their bodies were thrown into the bogs. They fell into the bogs. We're really not sure what happened to them, but they fell into the bogs, and they're perfectly preserved. They have a picture of one guy. You can see his whiskers.
Yeah.
And there's a rope around his neck.
Yeah.
So we don't know
Yes.
whether he was a slave with a rope around his neck, whether he's hung,
Yeah.
what the deal was with him. And a lot of the bodies are mutilated because they use a machine to cut the peat blocks, because peat's used as a fuel in these countries.
Yes. If you go to Ireland, you will smell peat because people burn it in their fireplaces.
Yep. Yeah, so that's another one, but I've really never found anything about DNA studies on them, but they are a mystery. They are definitely an archaeological mystery. And then we come to two stories that I really love. In fact, I start the ancient DNA lecture with the story of the Romanovs. And the Romanovs are the last ruling Russian family, and they were all murdered by the Reds, the Red Army, in 1918. What happened was the Bolshevik Revolution occurred in 1918, and the communists took over Russia. But at the same time, they were fighting World War I. And there were two armies. There was the Red Army and the White Army. The White Army was the Tsar's army. And the Russians really liked the Tsar, even though they didn't like him. You know, they had mixed feelings. They were very superstitious people. They didn't want to kill the Tsar. So what they did was they shipped him off to Siberia in the hopes that eventually, when there was a break in the war, they would send him to Finland and out through the what is that sea up there? No, it's not the Black Sea, the Baltic Sea, the North Sea. And he would eventually make it to France with the rest of his relatives. But what happened was the White Army was making inroads, and they were afraid that if the White Army won, that they'd put the Tsar back on the throne, and they didn't want that. So they hatched this plot, which they never told Lenin about until it was all over, where they actually herded the Tsar, his family, and all the servants into a room and shot them, bayoneted them, and then took them and buried them in mine shafts, and then took them out of the mine shafts and took them to other places. They poured lye on them and sulfuric acid, and it's just a horror scene. And of course, we have the whole idea about Anastasia. And even Disney likes Anastasia.
Everybody likes Anastasia.
You know Anastasia, the fake Anastasia.
Yes,
lived here.
Anna Anderson,
right?
Was it? Or that's what she
Anna?
called herself. I forget what her real name was.
Anna
yeah.
Anderson. Well,
Yeah.
that's what she called herself.
Yes.
But she lived in Seacliff.
Wow.
Long Island. Long Island.
Wow.
So I love to talk about the story. And then, of course, during Glasnost, when there was a lessening of tension between the United States and the rest of the world and Russia, they allowed people to come in. Of course, the locals in that part of Siberia, they knew where the graves were. They knew the whole thing. And so they allowed, I guess it was Americans, to come in. And it was in 1987, and they dug up the graves, and they found the remains. There were actually two graves. The Tsar, the Tsarina, and three of the daughters were in one grave, and Alexei, the son, who had hemophilia, by the way, and Anastasia were in the other grave.
And I didn't even realize the connect. I mean, I should have, but watching Victoria, the connection to Queen Victoria, and
she had a mutation,
yeah.
Victoria.
Yeah.
And she passed it on to one of her grandchildren, who was Alexandra.
Yeah.
Who married Nicholas, who was also related to the Kaiser and the King of England, the Kaiser of Germany and the King of England.
Right, yeah.
So, what a mess.
Yes.
So, at any rate, those were the two graves, and they verified that, who they were. And they reburied them. Actually, in my lecture, I have a couple of pictures of the reburial, and it's St. Peter. It's the big church, not in Moscow, but in St. Petersburg. I cannot remember the name of the church. But the funny thing is there's a mosaic on the wall of the entire Romanov family with halos. These are the people that they got rid of, and now they are enshrined as heroes. So at any rate, that's a nice historical perspective. But I really like that story because the whole thing with Anastasia, and
Rasputin is absolutely. I mean, he has been used in fiction. I mean, he's a terrifying and fascinating character as well, and we would totally go over time if we even got into that.
Yes,
but really, those are stories that absolutely, continue, and there are. Maybe it's because there's actual pictures of them as well that people are so attached to them.
They were beautiful.
They were beautiful.
Yeah.
Beautiful.
Yes. He was handsome. She was beautiful. The kids were beautiful. Rasputin was pretty ugly, but
he physically,
fascinating.
Fascinating, and physically embodies that, wizard, that evil wizard,
the monk,
yeah.
Mojik. The Russian word was mojik.
He really was Very interesting. But if we talk, like I said, if we talked about Rasputin, we'd be here for another
Well,
hour.
if you're really interested in that whole thing, there was a book called Nicholas and Alexandra. I cannot remember it was an American author, and he wrote the book because his son had hemophilia, and he was fascinated